All right, this video is going to accompany section 3.3 in the OpenStax textbook. So the nucleus and DNA replication. And I know in an earlier video, when you were learning about organelles, you heard a little bit about the nucleus. But we'll go into a little bit more detail here in this video. So take a look. Here's the nucleus. And typically the nucleus is near the center of cells, but not always. Sometimes it's pushed up against the plasma membrane. Sometimes it's in various places in the cell. It really kind of depends on what cell you're looking at. And we'll see that as we do some of the histology study in lab. In any event, nuclei always serve the same purpose. They house the chromosomes. So all of the DNA that you are born with is housed within the nucleus of each one of your cells. Every human being is born with 23 pairs of chromosomes, and so 46 distinct chromosomes. Now, you can't actually count 46 distinct chromosomes here, that's not the point of it, but each of these little squiggly lines here are meant to be those chromosomes. And so in the form of the chromosome, when it's really not getting ready for the cell to divide, we call the form chromatin, and it's just a real thin, spindly form of the DNA. And then as the cell begins to get to a point where it needs to divide, the first thing it has to do is completely replicate all of those 46 chromosomes so that it has a copy of 46 to go into one of its daughter cells and another copy of 46 to go in the other daughter cell. So there you got to replicate them. When you do that and go into cell division, the chromatin material condenses highly into what is known as a chromosome, and we'll see that a little bit later. So I'm going to hold off on that for a moment. Notice the structure in the middle is called the nucleolus, and the nucleolus is a structure in the nucleus that forms ribosomes. And the ribosomes actually are very important for protein translation. And the ribosomes, you actually see them out here on the endoplasmic reticulum. If you've already learned this, this folded material back here, back and forth, this membrane is called the endoplasmic reticulum, and those little golden dots on it are called ribosomes. I'll show you a little bit more about those in a minute, but anyway, the ribosomes are formed in the nucleus, the nucleolus, and they're shipped out of the nucleus and into the cytoplasm through these little pores called nuclear pores. Notice that the nuclear membrane that covers, that surrounds or bounds the nucleus is actually kind of cool. It's a two-part membrane, and it has an inner membrane and an outer membrane. You can see that all the way around. Both of these, the inner membrane is a bilayer of phospholipid, as well as the outer membrane being a bilayer of phospholipid. Pretty much exactly the same way the plasma membrane looks like. But one more interesting thing. If you come down here and look carefully, you will see that the outer membrane of the nucleus is the same membrane, see that, as the endoplasmic reticulum. They're the same membrane. And so really the endoplasmic reticulum is just a huge folding and interfolding and back and forth of the outer membrane of the nucleus. So a quite interesting connection there. Notice that the nuclear pores do not empty up into these cisterns, which are the folds of the endoplasmic reticulum, but instead they go directly out to the cytoplasm. That's really important because we're going to see that RNA is synthesized from our DNA molecules in here and shipped out of those nuclear pores, as much as the ribosomes that are made in the nucleolus are shipped out of those pores. So that's primarily the role of these nuclear pores, is to allow RNA molecules, we call them messenger RNAs, to get out of those nuclear pores and out into the cytoplasm, and also the ribosomes to go through them and get out into the cytoplasm. So we'll talk more about that in a minute. What I want to do is jump over to visible body for a moment. So in the visible body app, I want to show you just a little different view of the nucleus. So let's just open this up a little bit closer, zoom in, and take a look. What you see here are some of the chromatin bits that have not condensed yet. That's those kind of strings looking thing here, like your previous picture did. But notice that some of the chromosomes have begun to condense. And again, eventually there would be 23 pairs of these if all of them condensed. But let's click on one, and you can see that those are chromosomes, and those are condensed chromosomes when the cell is getting ready to divide. And so this cell is getting ready to divide. Here is the nucleoplasm. That's basically the cytoplasm, but inside the nucleus. Very similar kind of semi-fluid that has all the parts of the nucleus in it. And then this is the nucleolus we learned about earlier. Remember, this is what makes the ribosomes. And the ribosomes are going to be shipped out of these nuclear pores. Here you see them right there and out into the cytoplasm. Okay. And so the other thing that happens in here is that these DNA molecules will be transcribed into messenger RNAs, and I'll talk about that in a later lecture. And again, those RNAs are also shipped out of those nuclear pores like this right here. Okay, I want to show you one more thing. I want to jump over to the ribosomes just to show you. These are quite interesting. When the ribosomes, which again are in charge of translating proteins, that's what they do. They make proteins for the cell. Notice this picture is quite interesting because if you take a quick look, that's the nucleus, okay, inside here, the purple. This is the nuclear envelope. This is the nucleoplasm inside of here. Look how the endoplasmic reticulum makes this huge, mass outside of the nucleus. Very interesting. And this is a primary spot for protein synthesis. And you can see all of the ribosomes that are affixed onto those endoplasmic reticulum in order to do that protein synthesis. So if we click on some of these are all ribosomes. Click on that right there. Oh, I didn't quite hit it. There we go. Those are the ribosomes. But an interesting thing is to notice that the ribosomes also, some of them are actually not connected. Like look at those guys out here. Those are not connected to any ribosome. I'm going to see if I can get one. There you go. Just kind of free floating out in the cytoplasm. And you get those are called free ribosomes. They do the same thing. They translate protein, but just not onto the endoplasmic reticulum. A slightly different function, but interesting nonetheless. So those are the ribosomes. What I want to do now is I want to jump into back into your textbook, and I want to walk through very, very basic steps of DNA replication. And in order to do that, what I'm going to do is begin one more time with just an explanation of DNA. Now, if you remember in the chemistry chapter, you learned a little bit about DNA, and you learned that the nucleotides of the DNA are the monomers, and many of these nucleotides are put side by side or back to back to one another, and they create this very, very long string of nucleotides. And then with DNA, it's a double-sided molecule, so you actually have two strands of these nucleotides that are bonded together in the middle by hydrogen bonding. So here's one long strand of nucleotides, and you can see it just goes on and on and on forever. There's that chromatin form, and then as it condenses into a chromosome here. Anyway, come back out here. Take a look. There's those long list of nucleotides back to back to back to back to back, and they are base paired to another strand of nucleotides here. So it's a double-stranded molecule built into a helix, right? And so notice the base pairing. I think you learned about that in an earlier lecture, but notice that A always bonds to T, and T always bonds to A, and G to C, and so on and so forth. And so as you go down, you see that they're always the same. If I find a T on this side, it will be bonded to an A here, G here to a C here, always the same. Very, very important. So base pairing always works that way. If we come down and look at a slightly different view of the same molecule, we can take a look at the nucleotide. This is the nucleotide right here. It's got three parts, a phosphate, a sugar, and then the nitrogenous base. The nitrogenous base is what makes it either an A, G, a T, or a C. And those bond to one another by way of, as I said, by way of hydrogen bonding across. So here's a good example. Here's a thymine, and here's an adenine, and there's the hydrogen bonding between the two holding them together. And then the same goes here. Those are also, again, hydrogen bonding down here between the guanine and the cytosine. So that's just how they're held together. Not a big deal, but those are very easy bonds to break. And because of that, you can easily separate the two sides of the molecule apart from one another. There's one strand, there's the other strand. You can easily break those bonds and separate the two sides. And that's what's shown here. So just imagine that this strand, the top strand and the bottom strand here, were once bound together like they are here. See all the bonds holding them together? But we can have a molecule called helicase that easily breaks those hydrogen bonds and separates the two from one another. So as we take a look, these two have just been broken apart. the adenine here in red and the thymine here in gold have just been broken apart. And what that allows us to do is add a new nucleotide to the one that's previously there. So we're just going to base pair them. Notice here that this molecule called DNA polymerase is doing that very thing. So we've separated the two strands of the DNA molecule. Again, this is called DNA replication. There it is, DNA replication. And what's happening is this DNA polymerase is moving along and adding a new nucleotide to the previous one that's already there. So this green one is a cytosine. So it's going to say, well, I need to put a G with a C. So I need a guanine. And so it does. Grabs a guanine and puts it there in that spot. Here is an adenine, and it's going to say, well, I need to find a thymine and stick a thymine on there. What will we add here? That's a guanine, so I need a, hopefully you said cytosine. Here's a guanine. I need another cytosine. This is a thymine. What would I need here? Adenine, right? And so on down. I'm just going to keep adding the correct one to it. And when I get done, notice I've got a brand new DNA molecule that'll be exactly the same thing as the one it was previously. So you get two exact copies. And that's what you're trying to do. Just make a perfect replica of each chromosome. Because again, DNA replication is specifically getting ready for the cell to divide. Make two perfect copies of your 46 chromosomes. 46 to one daughter cell and 46 identical ones to another daughter cell. Okay. One last thing to quickly say is that notice that the molecule is getting replicated in two different places. Here, the top strand is getting replicated into what we call the replication fork. That's actually called the leading strand. There's a reason for that I'm not going to worry about at all. It's just too much detail for now. And look at the other side. It's going in the opposite direction. It's actually starting near the replication fork. There's a replication fork. And replicating in the opposite direction. And so we can see here, this is a guanine here that's getting ready to have a cytosine put next to it. And here's a cytosine here that's going to need a guanine. And here's a thymine here, sorry, an adenine here that's going to need a thymine, and so on. So you can do this work yourself. And this is called a lagging strand as it moves out in this direction. So again, a little more detail than I wanted to get into for that. But I think that gives us the information we actually need for this. If you go through and read a little bit about the initiation of the process, that's just breaking those hydrogen bonds and untwisting it, getting it ready with helicase. Elongation is just using the DNA polymerase molecules that you saw above. These guys right here, DNA polymerase, to lay down the new strand as a template of the old strand. And then termination. We're not going to ask you about how it terminates, but the process has to be terminated at the end. And that's pretty much it. I would highly recommend you take a look at some of these videos on DNA replication, and I think those are quite useful as well. And that's it for the nucleus and DNA replication.